Hello everyone, and thank you very much, Tomoko, for your very kind introduction. I'm really excited to be here, and I will tell the two discovery stories actually today from our highly productive collaboration between Schrödinger and Morphic Therapeutic, where we capitalized on Schrödinger computational platform and the excellence of the experimental execution at Morphic. I will start with a quick introduction of our collaboration between the two companies and how we worked highly synergistically, being in the different cities in the United States. I will spend most of my time today talking about molecule known as Morph 627, where I can show also the chemical structures of the compounds. And toward the end of the presentation, I will also mention the most advanced asset in the Morphic pipeline, the molecule known as Morph 057, and this is the selective oral small molecule inhibitor of alpha-4 beta-7, which is currently in the clinical trials for the treatment of inflammatory bowel disease, or IBD. And I will be happy to take any questions in the end. So just to set the stage, Morphic Therapeutic is the company which was founded to pioneer the oral integrin medicines using or leveraging their proprietary structural biology as well as the biology expertise. And a lot of this expertise has originated in the lab of their co-founder and the pioneer of the field of the integrin structural biology, Tim Springer. So integrins are the very interesting class or family of the heterodimeric cell surface receptors with a very unique ability to actually signal bidirectionally, inside out and outside in. So they also function as the signal transduction platforms. And these molecular machineries are highly flexible and they can adapt a number of different distinct conformations during this signaling process. And two of these conformations are shown on this picture. So one on the left is what's known as the band closed or inactive state of the integrin receptor, and the other one is fully open or extended open, which is also known as the active state. Historically, when people were targeting the integrin receptors, they were traditionally targeting them in this extended open or the active state. But the Morphic took on the challenge to discover and understand the pharmacology of the compounds that target specifically different states of this integrin receptors. So in addition to the conformational flexibility challenge targeting these proteins, which I just mentioned, there are additional two complexities associated with this target class. The first one is essentially achieving high levels of selectivity against closely related integrin targets. And this picture just shows all of the possible combinations which can happen between the pairing of those heterodimeric subunits. Some of them are known as alpha subunits and the other one is known as beta subunits. And as you can see from this picture, not all of the alpha and beta subunits pair up with each other, but there is very specific interactions which happen to form those heterodimeric structures. But nonetheless, there is about 24 different combinations of those alpha and beta subunits, which now you can imagine could possess a real challenge in the drug discovery campaign to achieve the designed level of selectivity against closely related targets while maintaining the right level of potency on target. Today, what I'm going to show you is the drug discovery program where we were interested in targeting alpha V beta 6 integrin, which is pairing shown here, while we were also needed to achieve selectivity against closely related alpha V beta 8 and alpha V beta 1. So the second important challenge is the optimizing compounds for these targets for their physiochemical properties, given the very polar nature of the binding site to generate essentially orally bioavailable small molecules to inhibitors for these particular targets. And I'll show you with an example why the complexity of this binding site comes into play. So with all these challenges, which I just highlighted, served as a great foundation for highly productive collaboration between Schrödinger and Morphic Therapeutic, where two companies were bringing highly synergistic expertise to facilitate this with this drug discovery efforts. So Morphic Therapeutic was responsible for all of the experimental activities such as including the compound synthesis, also executing on the very complex integrin assays, as well as they are truly the leader in the field of the structural biology for these targets, as I mentioned earlier. So all of this preparatory structural biology information which was generated by Morphic was really essential for us to enable our computational platform at Schrödinger to help driving this program forward, including our FEP plus technology. In addition, with Morphic being in the Boston and majority of the Schrödinger team spread between the New York City offices as well as Portland offices on the West Coast, we used and leveraged our enterprise informatics platform Live Design as a one-stop shop to be really efficient in the program execution. So we incorporated all of the data we obtained throughout the program in our Live Design platform. That started with all of the design ideas which were generated between the Schrödinger and Morphic teams, which then would progress into the modeling using our advanced tool, and all of the computational data as well as the structural biology data was kept within Live Design platform. And finally, after the compounds were synthesized and tested in the biological and NV-DMPK assays, it was allowed us very quickly to integrate all of that experimental data into the Live Design to continue our cycle, design, test, make a cycle. So this is the way we worked highly collaboratively between the two companies using Live Design as the platform. Now let me switch gears to the first vignettes on the compound Morph 627, which is the compound potentially targeted for treatment of the fibrotic disorders. In the interest of time, I won't go into a lot of the very complicated biology around alpha V beta 6 integrin, but it's suffice to say that the alpha V beta 6 integrin is implicated in the TGF beta signaling pathway, activation of which is associated through the downstream signaling pathway in the number of the potential fibrotic disorders. So the therapeutic hypothesis behind inhibition of the alpha V beta 6 target was essentially application for treatment of these fibrotic disorders. I also would like to mention, as I showed in the introduction, that alpha V beta 6, as all of the other integrins, have two potential conformations: closed conformation or inactive, and the open conformations, which is essentially associated with TGF beta signaling activation. And therefore, in the Morphic program, we're interested going after this closed conformation of this particular receptor. To also give away a flavor of the polarity of the binding site, this is actually the native RGD ligand, arginine glycine aspartate, which is a tripeptide. So it can give you a flavor of how polar the binding site is, and that possesses additional challenges when we're looking for the chemical matter for this target. So in the earlier stage of the programs, when we started working together with Morphic, they had a small preparatory collection from which team was successful at identifying weak hits and also generate some preliminary early SAR. So both of the starting points comparing the piperidine motif and pyrrolidine motif, we essentially equipotent on the target, so this is alpha V beta 6, and pretty much not selective against other target as expected for the early hit matter. So this is alpha V beta 1 and alpha V beta 8, which I will be talking throughout the presentation. I also would like to set a stage and orient everyone around the binding site of the alpha V beta 6 and in general alpha V beta family, where the alpha subunit, which is always the same, is alpha V is on the left hand side, where we have the bidentate interaction of the THN with aspartate 218. The right hand side is where the difference come into play. So the beta subunit could either represent beta 6, this is the desired on target, whereas the beta 1 and beta 8 are the critical off targets which we needed to achieve selectivity against. An SDL2 loop, which I will also be mentioning throughout the presentation, is the critical place where we felt that a lot of the potency and selectivity could be found by targeting it in a selective fashion. So one of the early discoveries in the hit ID stage of the program was incorporation of this phenyl group next to the carboxylate in both of the scaffolds. It was interesting because in the case of the pyrrolidine amide scaffold, it led to the significant improvement in potency. We were able to push the desired on-target potency to double-digit nanomolar level, and it did not actually have any impact on activity for the PP-ready named scaffold. And that was actually a very difficult thing for us to originally explain before we were able to fully enable our computational platform, including FEP plus technology. Because from the perspective of just docking the ligands into the active site, both of these structures containing this phenyl group with a pyrrolidine scaffold or piperidine scaffold will fit really nicely into the structure. And I will talk more throughout the presentation about the complexity of predicting potency and selectivity for this pocket. But nonetheless, this was an exciting SAR which team was able to generate early on in the program before we fully enabled the computational platform for the targets. So one of the significant downsides of both of the scaffolds was really poor permeability, which we knew eventually will lead to the essentially not having any oral bioavailability with these hits. So the first goal for this program was essentially optimize the permeability, which was measured less than 1 in MDCK A to B assay for both of these scaffolds. So in order to address this challenge, we look at the number of the amide linker replacements, and we did some SAR around essentially making the linear, exploring linear linkers such as just regular carbon chain or incorporating some polarity into the system. In addition, as we looked at some more constrained analogs such as the amide mimetics, as well as some more constrained systems with the additional aromatic system in the linker. So as you can see from the activity perspective in alpha V beta 6, we were able to essentially maintain the potency and in certain cases to even push it to the single digit nanomolar level. And what was more important, we now started getting the measurable or sufficient amount of MDCK permeability for these linkers once we removed the amide functionality. What was really interesting and exciting for us is that for the matched molecular pairs of compounds such as 5 and 6, when they had a very similar measured experimental log D, similarly to match pair between 7 and 8, where the log D was about 1.5, the level of permeability was different. And this was somewhat striking from perspective how we typically think about the permeability correlation with the lipophilicity. So we asked the question why. So at this point, we turned our attention into modeling of the 3D conformations of these ligands, and we hypothesized that the ability of these ligands, which are highly charged, to form this interesting intramolecular masking of two charged functionalities such as the carboxylate as well as the THN motif, is essential for improving the permeability of the compounds with more flexible chain. So you can imagine when this chain is not flexible, then the two ends of the molecule cannot reach each other as easily, and that was the hypothesis we generated why some of the more flexible linkers were more permeable comparing to another. So we were also able to show now in the systematic way that as we grow the number of rotatable bonds in this linker region, we essentially get better permeability for those compounds. So this graph shows the permeability as relates to the number of the rotatable bonds. And you can clearly see when we have a rigid linkers, all of them have poor permeability, but as we increase the number of the rotatable bonds to more than 8, we really get good chances of getting compounds in the good range of permeability, and eventually it would translate into the oral bioavailability. So we use this type of foldability predictions throughout the program as the proxy to compare compounds for their permeability potential. But the understanding of the flexible linker was only one piece of the permeability puzzle, because we were also able to identify that compounds which have essentially identical linker, so I'm showing the compound 9 here, which has now the pyrrolidine ether linker, and the compound 11, which just has an extra electron withdrawing group in this aromatic group, they have essentially the same flexible linker, but they have very different MDCK permeability as shown on the slide. So similarly, this compound which had a carbon linker had actually a similar MDCK comparable to the pyrrolidine ether. So at this point, what we really hypothesized is that it's a basicity of the spirulidine nitrogen, which now plays also an important role in the permeability of the compounds. So because this is the only charged species which is left once the intermolecular masking of two charged ends of the molecule happened through the conformation I showed you earlier. So we were able to establish a highly efficient and accurate method to predict this basicity of the pyrrolidine ether using the quantum mechanics method. And we were also able to show that the QM, when we predict the basicity of this pyrrolidine nitrogen of being less than 9, that's essentially what gives us much higher probability for the compounds now to be in this desired range of permeability. So we really needed to stay away from compounds which were predicted to be really basic and really work in this fairly narrow range of basicity of this pyrrolidine core to give us the compounds with the desired mix of properties. I also will mention that we didn't want to be in the close to the neutral range of the basicity, because that would give us other complexities such as poor metabolic stability, which we observed for the earlier compounds we made in this space, and they were also highly protein bound. So we really were operating in a fairly narrow range of basicity to combine multiple properties into one molecule. So we needed to have a very highly accurate method to predict this basicity of the pyrrolidine core as we were thinking during the prioritization of the compounds for the synthesis. So we were really excited with this method we established. So this was the QM protocol, which one of our modeling colleagues have developed and implemented on the program, where we were using it very actively to prioritize compounds for synthesis. We spot checked some of the compounds experimentally by measuring this pKa of the basic pyrrolidine using very accurate methodology. And we were really excited to see that there is a great correlation between the prediction of this basicity using our QM method to the experimentally measured pKa. And that also holds true for the range of basicity which we needed to be able to optimize compound for their permeability. So this good level of accuracy of mean anesthetic error of 0.25 was essentially really critical for us to be able to prioritize compounds. So after being able to figure out how to address the permeability of our early hits, we also needed to now push ahead and optimize those compounds I showed you earlier for potency and selectivity. We now established this pyrrolidine ether motif shown on the previous slides as the scaffold which we chose to progress into hit to lead and the lead optimization of the program. So FEP Plus technology, which I'm sure a lot of you might be aware of, is at the core of our computational platform and really allows us predicting the compound's binding affinity to the target receptor through the calculation of this thermodynamic cycle, which is shown on the slide. In addition to be able to predict compounds binding affinity to the target, desired target of interest, in this case it's alpha V beta 6, exact same methodology allows us to model the off-target selectivity if we would perform similar simulations using the crystal structures of alpha V beta 8 and alpha V beta 1. And that's essentially the workflow we utilized for this program and this collaboration, which really efficiently ran almost like a clock on a weekly or bi-weekly basis. So we would routinely collect all of the ideas and the live design from the joint team between Schrödinger and Morphic, and after all those ideas go into live design, our modeling team would first perform the FEP simulations on target using alpha V beta 6 crystal structures, as well as get a sense of the QM pKa predictions of basicity of the pyrrolidine amine, which I introduced on the previous slides, and only a subset of compounds which would pass these filters will gonna go into the in silico assessment of loss of binding affinity for the off targets, and eventually culminate in team selection and synthesis of the compounds which will pass through this funnel. So then once the compounds will get made and tested and analyzed, the cycle will essentially continue. And we run literally as a clock on a weekly and bi-weekly basis, which allow us very efficient optimization of the chemical matter. So overall, just to put things in perspective, overall throughout the program, we performed close to 3,000 FEP simulations looking at different alpha V beta 6 ligands. For about 10% of compounds, we obtained experimental data. As I will show you on the upcoming slides, we spent a lot of time really optimizing what we call as north substituent. This is the substituent which points into the SDL2 loop of these targets. But nonetheless, we were also interested in looking at some additional linker possibilities as well as the THN. For all of these three different places where we were doing the chemical changes, the FEP performance was highly accurate, well within the expected accuracy of the method, where we were able to essentially predict it with less than 0.8 PIC50 units for all of these three receptors. So this histogram shown on the left is one of the ways to look at the overall FEP plus performance during the course of the program. It essentially shows how many compounds fall in different buckets as it relates to the differences they predicted versus experimental potency. So the first two big buckets on the left essentially represent the compounds which were predicted within tenfold of experimental potency, which is an expected accuracy of the FEP method. And we were really happy to see in this program that more than 80% of compounds ever predicted and made in this program, we are essentially meeting this criteria of being within tenfold of predicted accuracy, and that essentially helped us to drive the program forward with this methodology. So, but it did require some important optimization of the method before we were able to get it to the level of accuracy as you just showed you on the previous slide. So in the earlier stages of the program, there was two potential vectors which we were interested in pursuing, starting from our compound 5, so pyrrolidine ether, which has a simple phenyl group, alpha to the carboxylate. So we were envisioning growing into this pocket created by SDL2 loop, either from meta or ortho position, both of which were accessible looking at the crystal structures. So we generated a subset of compounds in this case where we were looking growing into the pocket from meta position. So this is just represented from three structures shown here. Based on the predictions from our FEP model, we were able to show that we are driving the potency as we're growing bigger into the space, as represented by compound 12, 13, so predictions are becoming higher, as well as for this structurally different core where the pyrrolidine is directly attached to the phenyl group. Unfortunately, even though we were able to achieve some additional boost in potency from the compound 12, where we had a small hydrophobic substituent pointing into the SDL2 loop, as predicted by FEP, for the bigger substituent and more diverse substituents, we were not necessarily gaining any additional binding affinity. So essentially, the binding affinity was either staying flat, or we were actually losing affinity comparing to the predictions. So overall, these two compounds might represent what we would call as a large outliers from FEP plus performance, being 2 log units off from our predictions. But what's really important, and why I really wanted to show this example, is we did not give up getting this experimental result. Our computational colleagues used this result to understand and do the tweaks to the computational model to be able to come up with the next generation of our model, which would allow to make for a better and more accurate prospective predictions. Very briefly, there was a specific flexibility of the SDL2 loop, which needed to be taken into the account appropriately during the FEP simulation. And once our computational team has incorporated those changes, we were now able to accurately recapitulate the experimental data with the computational method. And that was really important for us as we were progressing into the active optimization cycle to have a fully performing FEP model at our disposal. Okay, so the other place at the same time when we were looking at the SAR expansion in the ortho position, we were more successful in finding the alternatives for the cyclopropyl group. Similarly to the meta prediction, to the meta substitution, if you put a small hydrophobic group you can drive the potency to about tenfold to the single-digit nanomolar level. But also what's interestingly, in this case, we were interested in expanding this group either by growing into the bigger cycles such as cyclobutyl, or more in the linear fashion, sampling how much space we have under the SDL2 loop. So looking at these two chemotypes, so in both cases, the FEP was accurate in predicting maintenance of potency. We essentially were not driving the potency to the next level. We were not quite pushing the low single-digit nanomolar potency where we needed to be for this program to be successful, but nonetheless, those two substituents were tolerated. It was also interesting that in this case, we were able to start seeing the reasonable selectivity against the closely related off-targets, such as alpha-V beta-8. So I mentioned that the beta-8 target has differences in this SDL2 loop. So as we were growing into that space, we were getting some selectivity. The challenge with this series was really poor PK, as one could imagine. So with this exciting data points, and also after having fully optimized FEP Plus model, we undertake on the campaign where we were interested in exploring this chemical space very broadly, and then prioritizing compounds based on the FEP model to really push the level of potency about tenfold from where we were observing it for these current compounds. It was important to have FEP model at our fingertips, as we were pretty creative in generating thousands and thousands of different ideas and then prioritizing them through the funnel, as I introduced earlier. So one of the most productive and exciting explorations was looking at the subset of THF and THP substituted analogs, which were connected through 2, 3, and 4 positions to the phenyl ring. So we started with our compounds and then essentially connected all of these isomeric structures to the phenyl ring, and we did the FEP assessment. It was out of all of these possible isomers, which I'm showing on this slide, including closely related isomers, which are just different by the stereochemistry, FEP had clear preference for 2-THP as isomer as shown on the slide. So all of these numbers represent FEP prediction before the compounds have been made. So it was very interesting result. It was also not obvious, as there is no specific interactions which we observed to the protein backbone during the simulation. And I think we can all appreciate that from the size perspective, all of these ligands would essentially occupy binding pocket in a very similar way from size perspective. Nonetheless, this was an exciting result, and the team didn't shy away from working out the complicated chemistry to make this compounds. And we were really excited to see that it panned out experimentally exactly as we had predicted. So we have followed up with the compound 17. So we made compound with the 2-THP substitution with the S stereochemistry here. We also introduced the fluorine atom, because that was critical for us to achieve the proper amino acid pKa of the pyrrolidine amine, as I mentioned earlier, to drive it below 9 to also achieve the permeability in the assays. We were really excited to see that we now essentially prediction was really similar to what we obtained computationally, and this compound was now pushing the bottom of our biochemical assay on target of alpha-V beta-6, which was just around 1 nanom. So we were really excited for this breakthrough in potency, which we were able to identify using the FEP simulation. And Bryce Harrison, who was my counterpart at Morphic, was really excited, saying, yes, now FEP absolutely can drive the program forward. So with the compound 17, it's essentially became an important in vivo tool compound for the program, showing pretty favorable profile, PK profile across the species, and also an acceptable level of selectivity against both off targets, alpha-V beta-1, alpha-V beta-8. So we're at about 50-fold for this starting point. It was also exciting for us to generate crystal structure, co-crystal structure of the compound 17 with a receptor, alpha-V beta-6, to just confirm how non-obvious it was that the effect of the specific isomer of the 2-THP motif, which didn't pick up any interactions within the SDL2 binding pocket. So it was really an interesting finding which was driven by ideating a broad chemical space and then exploring it with FEP plus technology. In addition, this compound had a pretty good MDCK, as we predicted from modeling and making sure that the pK of this nitrogen is the desired space by the fluorine substituent, and eventually led to the acceptable level of oral bioavailability as shown in both red as well as the NHP. So in the next stage of the program, we set a very high bar, and we really needed to push this level of selectivity for the related targets alpha V beta 1 and alpha V beta 8 to the next level. And our goal was to essentially continue and exploring expansion into this pocket to improve the selectivity. So we used the exact same funnel I introduced earlier, but we put a very stringent criteria for the compound selection. So now we were really interested in compounds which were at least had a predicted PIC 50 of more than 9 when modeled on the alpha V beta 6. And when we were modeling the beta 8 and beta 1 selectivity, we were selecting compounds which were predicted to be at least 100-fold selective. So this slide shows you again one of the very exciting and very quick and productive ways how we were able to take our pretty advanced lead, compound 17. So it had really good activity against alpha V beta 6. It had reasonable selectivity, but we wanted to drive it to the next level. And in this particular case, we did the systematic gem dimethyl walk, where we introduced gem dimethyl substitutions at different positions around this THP ring, whether it's connected at 5, 4, 3, and 2 position. And then we ran all of those compounds, including their SNR isomers, through the FEP simulations. And we were looking for the compounds which would at least maintain, if not drive the potency to the next level. And we were really excited about compounds which would be at least 100-fold predicted selectivity on both of targets. So there were two cores, two particular chemotypes which came close, 3 substitution and 4 substitution, with 4 substitution being our favorite, and that's the first compound we prioritized for synthesis. The compound 3 also had a pretty reasonable profile, which we were also excited to test. Maybe not quite at the level of alpha V beta 1 selectivity, where it was predicted to be similar to the reference. So when the substitution happened at that 5 position, we didn't see any improvement in potency, neither selectivity, and substitution at 2 position essentially led to potency and selectivity loss. So fast forwarding, after we completed the chemistry here, we were really excited to see experimentally that the compound 19 indeed meet all of our criteria in terms of now completely pushing and bottoming out the assay in alpha V beta 1. I'll mention about the cellular readout in the upcoming slide. In addition to that, these compounds now showed really high level of selectivity against alpha V beta 1 and beta 8, which were essentially nicely selected based on the FEP simulations. So the compound 20 also was pretty reasonable overall profile, but not as high levels of selectivity, again showing consistent trend with what we observed computationally. Out of scientific curiosity, we made compound 18, where we put substitution into this position of the THP ring, and as we predicted computationally, this compound did not give us any boost in potency and was really not selective comparing to the reference. We were not interested in pursuing compound experimentally, which was predicted to lose potency as well as losing selectivity. So with compound 19 in hand, we only needed to do 2 more modifications before we arrived to our developmental candidate, MORPH 627. So we tied gem dimethyl group into spirocyclopropyl, and we also introduced the fluorine atom into the aromatic ring for the trick which I described earlier, where we can drive the basicity of this pyrrolidine motif from 9.1 to 8.6, therefore helping with the permeability of compound and really pushing it into the highly desirable range, which then in turn led to the really great oral bioavailability. In addition to that, I also want to mention now the other assay which was really critical for us. So in addition to measuring this compound's selectivity in the biochemical assays, we were also measuring them in the ligand binding assays in the presence of human serum to assess their cellular selectivity as well as potency. So it was really exciting. This was one of very few compounds where we really saw very small shift between the biochemical or binding affinity to the cellular readout, which was run in the presence of human serum. So you can envision that this is much more elaborated assay, which now takes into the account the protein binding component, and we actually only saw a small shift from our binding affinity to the fully corrected protein binding adjusted cellular readout. What was also important for us is that we maintained high levels of selectivity in the cellular readout for alpha V beta 1 and alpha V beta 8. And finally, compound MORF627 had a reasonable profile across the species, so I'm showing you half-life in rat and as well as in the NHP. So with this compound in hand, our DMPK team did the full human dose prediction based on the allometric scaling. So this compound was predicted to be efficacious at 100 milligram QD dose, and that was covering the full IC50 plasma corrected cellular assay, which I just introduced to you on the previous slide, at trough, while not touching any of those off targets which I mentioned. Alpha V beta 8 is shown here, and very similar if you look at the level of engagement, we are not touching the alpha V beta 1 as well. So this is the overall predicted profile for the human. I will also mention that this compound was actually efficacious in the bleomycin-induced fibrosis model of IPF, where it was done at 3 milligram per kilogram dose, and it showed similar efficacy comparing to the reference compound which was tested side by side with this molecule. So overall profile of MORF627 was supportive for its advancement into the IND-enabling studies and GLP toxicology. And just in summary, I would mention that this joint Schrödinger morphic therapeutic efforts led to the discovery of the highly potent and highly selective and orally bioavailable alpha V beta 6 inhibitor. We used the combination of traditional medicinal chemistry methods supplemented with large-scale FEP and QM methods, was capitalized on the Schrödinger platform. Unfortunately for this program, further development of this particular compound was stopped in the later stages of the IND enabling studies due to the suspected on-target toxicity observed during the non-human primate safety studies. So it was an unfortunate outcome for the beautiful compound and the program which we ran together with Morphic Therapeutics, but all of the learnings we gained during this project, we continue applying up to this date on all of the joint program within our collaboration. So there is a lot of great lessons learned and a lot of ways how we can model this very complicated integrins. I think with this, I'll just want to acknowledge the huge team of people who was working on this particular program from chemistry, structural biology, pharmacokinetics, screening, biology, as well as the Schrödinger computational and medicinal chemistry team. In particular, would like to thank Bryce Harrison. So he was my counterpart and project lead of this program at Morphic Therapeutic, and we worked really well together to this successful discovery of this compound. I think in the last couple of minutes, just to make sure that we leave some time for questions, I'll switch gears and talk very briefly about another compound which we co-discovered with Morphic Therapeutic and within Schrödinger collaboration. And this is for selective oral small molecule inhibitor of alpha-4 beta-7 inhibitor. And since this webinar series is into the clinic, I thought it would be really good to mention this compound briefly. Unfortunately, I won't be able to show you any structures for this compound. As you understand, this is now in the active clinical development, but I can share you the path how we got into this compound. So alpha-4 beta-7 is a very well proven mechanism to treat the inflammatory bowel disease. There is an improved antibody, vedolizumab, which is an injectable to treat this indication. And this program, when it started as the part of the collaboration, was essentially envisioned as the modality shift, where our goal in the collaboration was to discover the oral small molecule inhibitor, which was also highly selective for this particular target. So I'm not going to go into the biology. I'll just mention that essentially it's well-established mechanism, as I mentioned, and this particular antibody inhibits alpha-4 beta-7, which is on the surface of the T cells from trafficking to treat ulcerative colitis and Crohn's disease. So it's also expressed majorly on the gut-associated endothelial cells. So the real challenge, as I mentioned here, was similar to the program I described earlier, where we needed a high level of selectivity. So in this particular case, the selectivity needed to come for alpha-4 beta-1. Now, again, you can envision that the alpha subunit is the same, but the beta-1 and beta-7 are different in this pair of receptors. And this is, again, possessed another design challenge for the team to make sure that we can target only this pathway selectively without bringing the potential risks of PML, or progressive multifocal leukoencephalopathy, which was known as the risk for the earlier compounds which were not selective against alpha-4 beta-7. So in general, the small molecules alpha-4 beta-7 inhibitors are known in the literature, which are potent, but it was really difficult to bring potent, selective, and also compounds which had a good PK profile. And this is what we worked on together with Morphic to bring into one molecule. Again, we very robustly capitalized on the synergies between the two companies. So Morphic Therapeutic in this case was able to provide us with more than 100 proprietary crystal structures which actually ranged from alpha-4 beta-7 to alpha-4 beta-1, and we brought it together with a Schrödinger computational platform, including FEP technology, which I explained with the previous case study how we can use for optimizing the selectivity. So after running this program very collaboratively, we were able to achieve remarkable success, making the compound which was not only potent on the alpha-4 beta-7 on target, but we were actually able to achieve the desired level of selectivity against alpha-4 beta-1, the key off-target I mentioned, on the same order of as the approved antibody vedolizumab. So I mentioned earlier that there are some other compounds which are also potent, but they are not selective. So we were the first ones to be able to jointly co-discover the compound, which was a small molecule, orally bioavailable, selective inhibitor, which is now progressing through phase 2B clinical trials, and very recently Morphic announced some exciting data from phase 2A on this compound. So with this, I think I'll finish and stop here. I'd like to thank you, all of you, for your attention, and we'll be happy to take any questions.